Hello, my name is Gavin Ockert, and this is my presentation on the Environmental Project, which was completed under the guidance of Professor Paul Richardson. The Environmental Project focuses primarily on bacteriophages. Bacteriophages are viruses that parasitize specific strains of bacteria, often killing the bacteria while doing minimal damage to the surrounding environment. The bacteriophage attaches to and penetrates the surface of the bacterium, injecting viral DNA into the cell and allowing it to reproduce and form new bacteriophages inside the cell. These bacteriophages lyse the cell and are released, resulting in cell death. It is estimated that there are approximately 10 to the 31st bacteriophages on the planet, making them more common than every living organism combined, including bacteria. As a result, bacteriophages can be found virtually anywhere in the world, from estuaries to deserts. And because of their ability to seek out and destroy a certain strain of bacteria without causing significant damage to the surrounding organisms, bacteriophages are currently being considered for use in fighting bacterial outbreaks and bacterial infections, as they can eliminate threatening bacteria without serving as a danger for humans. So, the two primary purposes of the environmental project are to Number one, determine if there are bacteriophages, or coliphages, which are essentially just bacteriophages that attack coliforms, that kill strains E. coli K12 and E. coli B in any of Coastal Carolina's aquatic testing sites. The second purpose of the project is to try and identify these bacteriophages' viral families, which would allow us to potentially isolate and perform further testing upon the coliphages that we find in these water systems. We began our process by collecting water samples from 11 different aquatic testing sites from around Coastal's campus, then filtered and amplified these samples, which allowed any bacteriophage in the water samples to reproduce, making it easier to test for. We followed this up by performing plaque assays with each amplified water sample using plates spread with strains E. coli K12 and E. coli B, which helped us fulfill our first purpose by revealing whether or not there were bacteriophages that kill either of the two aforementioned strains of E. coli present in any of the water samples. Here on the left is an example of a positive plaque assay. You can see that the majority of the plate is clouded, and that's all bacteria, which in this case it's E. coli strain B. And in these circles here, we would drip amplified water samples, in this case from site 9. This example tested positive because the bacteria is completely gone in these areas as it's been killed by bacteriophage. A sample was considered, quote, non-determinate when there was a separate growth in the plate that made it difficult to tell if bacteriophage was actually present in the sample or if the results were just unclear. In this example, you can see right there, it looks like there's some sort of bacterial growth other than E. coli right on where we dropped our water sample. And so for that reason, we're unsure if bacteriophage was actually present in the sample or not, and so we just have it marked as non-determinate. A sample was considered negative when there was no contamination and when there was no effect on the bacteria in the sample regions. When you look at the circles here, it just looks completely blank, and so that's how we knew that it was negative. Using our plaque assays for reference, we then performed DNA extraction and PCR on the samples that tested positive or non-determinate for phage. Our primers are shown in color coordinated on the left, and as you can see, we use 3 different primer sets for our PCR. Finally, we followed the PCR up with a gel electrophoresis using our 3 different primers, and then we documented the results. This is an example of what one of our gels would look like after it had been completed and photographed. In the bottom right here, you can see a good example of what a positive lane would look like for the positive control, especially in the quote O primer section, as there are bright white bands that run directly parallel with the latter, which is what we want because that's our positive control. If you look in the other lanes here, you'll see that there are no bands, meaning that in B9, K6, and K8 in this example, they're negative. You can also see this with our negative control, which is what we want in this scenario. So this is a good example of what a typical gel would end up looking like for us. Here on the right is an overview of the 11 testing sites that we collected from numbered. Sites 1, 2, 3, 4, 5, 6, and 8 were all located in public ponds around campus. Site 7 was a small drainage ditch near the woods that contained mostly rainwater and runoff. Sites 9, 10, and 11 were all located along a larger, more central drainage ditch with tree coverage. One important thing to note here is that the drainage ditches were not treated with any antibacterial measures, whereas some of the other sites may have been treated. This would have an effect on our results later on. So here are our results. We collected for a total of 13 days spanning from June 3rd to July 26th. You can see on the left here that our positive plaque assays were most common in sites 9, 10, and 11, which remember were all located along an untreated drainage ditch. And then you can also see that we had some positives in 1, 2, 6, and 8, but the main takeaway here is that bacteriophages that kill strains E. coli B and E. coli K12 were most heavily present in sites 9, 10, and 11. On the right, you can see that this was pretty consistent with both strains of bacteria, with bacteriophages that kill E. coli B being slightly more common in our results than bacteriophages that kill E. coli K12, but still pretty consistent across the 3 sites. We can see here that the non-determinant tests were pretty spread out across the testing sites, with the main exception being site 10, which had the lowest temperature of non-determinant plaque assays since that site was usually positive. You can also see with the pH that sites 9, 10, and 11 showed much lower pHs than most of the other sites. Remember that these were located along a drainage ditch, which was likely not treated. Also remember that site 7 was also a drainage ditch, albeit much smaller and composed primarily of rainwater, and was also untreated and has a much lower pH than the average. You can see a pattern here between the untreated sites 9, 10, and 11, with lower pHs having a higher number of positive plaque assays, suggesting a possible correlation between treatment and bacteriophage presence. And then on the right here, you can see the average temperature of each site, with the latter sites having a lower temperature than the former sites. It is easy to try to draw a correlation between temperature and bacteriophage presence, especially since the sites with the highest number of positive plaque assays had the lowest temperatures, but this is likely a red herring. The drainage ditch whose sites produced the most positive plaque assays was shaded by trees, which could have led to a lower temperature than the ponds that face direct sunlight. Additionally, site 8, which had very few positive plaque assays, which was much deeper than the other sites, which could have led to lower water temperatures, again suggesting that there is little correlation between temperature and coliphage presence. What can we conclude from our results? Well, for starters, we can see from our findings that there definitely are coliphages in at least some of the aquatic testing sites around coastal. We had a high enough number of positive plaque assays to determine this, with most of them being in sites 9, 10, and 11. As I mentioned earlier, our results suggest that we are more likely to find bacteriophages in sites located along a drainage ditch. This could have been as a result of the fact that the drainage ditches were not treated, where some of the other sites may have been. It also could be because E. coli is more likely to be present in drainage ditches because of an increased concentration of fecal matter. Greater amount of E. coli may have attracted more coliphages and thus could have resulted in a higher number of positive plaque assays for sites located along a drainage ditch. Overall, our findings suggest that there are coliphages in coastal water systems and that these coliphages could be found more frequently in drainage ditches. So, how can we use this data that we collected and what does it mean for the future? Well, one potential use of this study would be to use bacteriophage to control bacterial blooms in public. Our findings suggest that bacteriophages found in nature, in places that we walk by every day, are capable of killing harmful bacteria like E. coli. When applied to a larger setting, bacteriophages could be taken from the wild and used to control bacterial blooms of E. coli without endangering wildlife or humans. We can also take what we learned and build off of it in future studies. In the future, we could use more strains of E. coli than just E. coli K12 and E. coli B in the pursuit of a bacteriophage that can effectively kill many different strains of E. coli, making it useful for things like public bacterial control that I mentioned earlier. More primers can also be included when performing PCR. Though they tested positive on our plaque assays, the bacteriophages that we found did not match any of our primers. In the future, we can include more primers to increase our chances of finding and isolating bacteriophages that are effective in killing different strains of E. coli. Overall, our results have suggested that some coliphages are capable of effectively killing different strains of E. coli, giving them potential applications in slowing bacterial blooms in nature to protect the public. And our study can be expanded upon in the future by testing with more strains of E. coli and by using more primers. I would like to thank Professor Paul E. Richardson for allowing me to mentor under him during this project. I would also like to thank Madison Gentillo for training me and providing me with the skills necessary to effectively carry out my procedures. Finally, I would like to thank Ryan Carter for compiling our data into digestible graphs and for providing me with photographs throughout the process. Thank you for your time and attention. This concludes my presentation. Necessary references for all images will be included in the next couple of slides.